Hello, everybody, and welcome to the June 19th, 2025 Inventors Gathering, which occurs every third Thursday at 8 p.m. U.S. Central Time for Public Invention. We have a number of speakers usually. Today, we're very pleased to have Dr. Avinash Baskaran, who recently completed his PhD on the subject of modeling neuromuscular fatigue and weakness in the human hand for people with various disease conditions as it relates to robotics in particular, with the data-based solution. Recently, Public Invention held a fundraiser and demo day in Austin, Texas, which was partially successful. We'll be talking about that in future episodes, and we invite other people to join us. Public Invention is a U.S. 501c3 public charity, so your donations are tax deductible, and we do need your donations as a show of support for Public Invention and our work. And now let me hand it over to Avinash. Take it away, Avinash.
Thank you very much for the warm welcome. It was great to participate in the gathering that we had over the weekend. And so that was a really great experience. And thank you all, kind of getting to see some of those familiar faces back again. So great to keep connecting with people and excited to continue. I probably should have put emails down here, but feel free to reach out to us and talk to us more about this as we go forward. And if you have any questions in the audience, just chime in and interrupt me, and that's totally fine. So today, I'll be talking a little bit about microscopy, specifically how it plays a role in traditional clinical diagnosis, and the shortfalls of not having microscopy in rural or places where that kind of technology is not easily maintained or easily operated by people there. And I'll talk a little bit very briefly about how this fits into a larger strategy for a device or a system which may allow for ubiquitous medical sensing and diagnostics on demand rapidly in any location. So something that we're calling the Librecorder. Yeah. Okay. So common clinical uses of microscopy, just to give an overview. So things like urinalysis, so identifying cells, bacteria, and the byproducts of bacteria in urine. Hematology, so using blood products, including blood smears, to characterize diseases that might affect blood cells. Classical microbiology, so things that may exist inside the body or in the environment. So things like bacteria, fungi, and parasites. So cytopathology, so some of the more kind of life critical applications. So kind of detecting cancer cells and trying to characterize some of those more significant things. So histopathology, kind of a very similar vein. So trying to characterize clusters of cells and clusters of biological material, which may indicate diseases like cancer. An interesting topic is semen analysis. So there's obviously the application as it relates to humans of wanting to understand and take control of your own health. And as I'll talk about it later in the presentation, there's also the interesting application related to related to animal semen analysis. So why is your bull not producing as many or of such cows that you desire, and so forth. So microscopy is really great, and it allows for a wide variety of biological characterization and diagnosis, which is really its own topic of discussion. But some of the problems that arise, as I mentioned, is when simply the access to microscopes is limited. In the world, 3.5 to 4 billion people lack access to essential diagnostics, and microscopy is one of those that often gets left off of the list in primary care settings because it's something that can take time to yield value in diagnostics, and it can also take specific expertise in usage and maintenance. And so those are just things that are not always easy to afford, especially in, as I mentioned, communities where supply chain logistics may be strained, or places where there's not traditional kind of infrastructural support in terms of training clinicians and training lab workers and technicians to use these things. The World Health Organization lists microscopes as an essential for medical clinics, but again, many facilities lack it. And so the results of that gap in access to microscopy is a significant number of deaths due to a lack of essentially early diagnostics. So you really want to catch some of these diseases like cancer as early as possible. And people who are getting labs only, maybe every few years, there's going to be things that slip through the cracks. Whereas, having early diagnostics through access to microscopy can make a significant difference in catching those things early. So these include things like tuberculosis and cancer, malaria, all things that can be assessed through microscopes and play a significant role in those uh, less uh, less resourced areas. And so delayed diagnosis can yield many preventable deaths. So things that could be caught early in the process and treated, but might slip through the cracks otherwise. And so these preventable diseases can be addressed with improving access to microscopy. An interesting point we discussed a little bit earlier in the talk is the implications for the U.S. So not only, we think of the U.S. as a place where most medical challenges can be addressed to the highest degree that is currently possible anywhere. Like it should be as good in the U.S. as it really can be anywhere because, well, we have such a robust supply chain and a lot of domestic manufacturing and funding for addressing such problems. But there are many rural areas in the country, especially in rural Alabama, where I'm based and where I'm conducting this research, where hospitals, community clinics, and small community health centers may lack, urgent care centers lack robust diagnostic equipment like microscopes. That's just not something that they usually utilize in their daily practice. And people have to travel significant distances to get labs done. And so, for one example, I think it is the case that there are millions of women in Alabama who live more than 30 minutes away from an OBGYN. There are many places in Alabama, many counties, I think 40% of counties which don't have a dentist. So there are significant challenges like that, where even in the U.S., there's a significant lack of some of these things. An important application that we've been discussing is cervical cancer. So a lack of access to microscopy means these really deadly and devastating cancers just cannot be caught early. And rural counties in the U.S. Have the highest cervical cancer mortality and higher mortality in a lot of the cases that we mentioned where microscopy can play a significant role, despite the availability of national programs which may be able to solve these. Let's see, is this okay? And so to talk a little bit more about the need for microscopy in the U.S., we talked a little bit about cancer diagnostics, particularly those things that can be assessed with microscopy through essentially diagnostic labs. An interesting use case is migrants and people moving between vulnerable communities. And so these are people who may be carrying diseases from places where they don't have labs and good healthcare infrastructure, and they may be moving to places where even though there is good healthcare infrastructure, they may not know to be pursuing labs and so forth. So the idea is to say, even if you're moving from the rural South, we want to have those resources available everywhere. We want them to be ubiquitous. We want people to be able to screen themselves on demand at any time so that they're not at risk because of migration to these types of things. An interesting statistic is sepsis. So this is a sepsis from UTI and other infections, something that could be diagnosed through microscopy, which leads to over 250,000 deaths in the U.S. So smartphone-based microscopy and field deployable microscopy is an interesting field because it is trying to reduce the barriers and the cost and the complexity of microscopes so that they can be deployed anywhere and be a more ubiquitous part of healthcare and kind of self-service on-demand healthcare. These are low cost and often scalable to manufacture. Again, they provide that rapid point of care diagnostic capability. They enable telepathology, so someone who is living in a place where the road access is not very robust, or otherwise the supply chain is not very robust. There may not be doctors in that area specialized to treat or diagnose a specific injury, but having these you know, telemedicine tools can enable pathologists from all over the world to consult together. One of the interesting use cases I've been reading about is field deployable microscopy for connecting and consulting the network of clinicians in a country or across the globe on one case. What an exciting opportunity for the best doctors in the world to be treating one case together anywhere in the world, right? Kind of one of those sticky topics, but interesting to discuss is integration with modeling. So that can be AI and machine learning, or just building databases that can be used in analytical models as well. And so one of the larger goals of our project is to promote health literacy and accountability. So giving people the tools that are ubiquitous and can provide very high quality diagnostics on demand will even though they don't take even if they lack the capacity to take action and treat themselves. Obviously, they're not always doctors using these types of things, but just to know what's going on with your health and provide that level of self-information is, I think, a big step forward from folks not having any sense of what's going on with their health. And so we're hoping to give people more accountability and more literacy, more access, and more control over their health. And so one of the interesting and compelling things about new development in any field is that it invites participants, invites participation by communities. And so we are hoping that by pursuing a community-driven ideal of field deployable technology, rapid distributed manufacturing, that we are promoting engagement by healthcare communities, rural communities, physician and medical staff, kind of working together to fabricate, upgrade, and participate with one another in this community-driven R&D process. So I think something we haven't quite addressed in our project together, but I think it's something that I've learned and kind of seen in stark contrast this week in New York for the open source events, is the distinction between mobile microscopy and benchtop microscopy. And both can be smartphone based and both can also be open or closed source. So there's a lot of kind of overlapping bubbles here. So if you talk about a benchtop smartphone-based microscope, it can stream to a smartphone. So it doesn't have to be mechanically connected to a smartphone to still really be a smartphone-based microscope system. So there are a lot of kind of open questions in how should we think about smartphone-based microscopes? So one of the interesting examples we've been studying is the U-scope or the microscope, which is a smartphone case, a smartphone attachment, which attaches to a smartphone case, and it includes a lens assembly and a light pipe to transmit light from the flashlight on a smartphone as a kind of a illumination for a slide. And so this is something that can be used. Right now, it's tailored for the iPhone, but in theory, it could be tailored for any mobile device, which has a camera and Wi-Fi antenna to take pictures, transmit them to a central repository, conduct diagnostics, and so forth. And so the experiments that have been conducted by this group include complete blood cell counting, which is a standardized test and clinical procedure. So this is something related to kind of holistic analysis of blood cells and blood imaging. And an interesting experiment that they conducted was to identify and count red blood cells. And so this is a field of study that might be called hemocytometry. And it populates a series of tests and analysis and diagnostics related to that measurement. And they were able to, with high precision, count blood cells and characterize their shape. And so this is kind of a step forward. So we see that smartphone-based microscopes are there are not as fundamental design limitations. They're capable of taking images. They're capable of being magnified to such a resolution that they can take high precision images of blood cells. and that they can do perform autonomous counting at the same similar level of precision as human counting. So they were able to produce essentially an autonomous hemocytometer uh, with some not as great as a human doing it, but kind of they were able to take that first step there. Let's see. So another interesting thing they were able to do is visual confirmation, as I mentioned, of blood cell size and shape. And I just want to mention that this is part of a larger ecosystem of hemocytometry and blood cell characterization. So things like anemia, um, and sickle cell disease and things like this, where the cells may exhibit visual characterization, visually characterized defects and morphology. So these are things that large models and distributed databases may be really effective at catching early in the process, especially with self-screening and on-demand screening. And so we talked about that there might be an interesting distinction between mobile microscopes and benchtop microscopes. So a very important open source microscope that's been developed is the open flexure microscope. And so I was very fortunate. I was actually able to meet with Julian this week in person, and apparently he often carries around an instance of the open flexure microscope with him. And so he set it up for me in a pub downtown, and we were able to, I was able to see it work, and it was really quite an experience. So it has a Raspberry Pi, connects through a Wi-Fi router repeater device to an iPad or any device. So it streams to a web-based platform, and you can control the microscope through the web-based platform. So it's a really cool thing. And one of the interesting things that he mentioned was a hospital may be really large, and it may be difficult to kind of schedule time with the pathologist or to get him, him or her down to your space, where this microscope may be based or might be moving around. And so a pathologist can control this remotely from his smartphone or mobile device, and provide simple instructions to the operator. In fact, and the motors you can see here can offer self-alignment, self magnification and adjustment. An interesting thing it does is it's able to move its field of view, so it can take pictures and stitch them together. So a very useful thing they're able to do. So some of the experiments that have already been conducted with open flexure microscope include malaria diagnostics with really decently high accuracy. Automated autofocus again for that hemocytometry blood capturing. Let's see. So multimodal imaging. So kind of having a standalone microscope platform can allow you to pack a lot of cool hardware into it, while still retaining some of that smartphone freedom of being able to access, control, and store data and stream data back and forth from a microscope platform. And they've also been able to do studies in histopathology, and as I mentioned, that kind of semen motility analysis, which is relevant for humans and animals and really a wide variety of interesting cases. So it's great that there are numerous field microscopy solutions. But one of the problems is that these haven't really thoroughly been evaluated with regards to how they might integrate and complement existing clinical workflows. So what I mean is doctors in the field who might take advantage of these things, especially maybe in rural or under-resourced areas, it's not really clear that such devices are readily applicable to their existing workflow. So for example, we have this smartphone-based system. We've talked about in this talk, maybe some of these microscopes could be useful for characterizing animals or characterizing things like skin samples, like without the need for extraction because it's a smartphone, right? But it's not really clear that that anyone is well trained in using a mobile handheld microscope for thorough diagnostics. And that's certainly, I think, not a significant part of existing clinical workflows. So, and then when you talk about using that for animals, like you're going to hold a chicken still in your hands and take pictures, like it's a really challenging problem. And specifically, it's almost an industrial and systems type of problem, which is to say, are the engineering requirements currently specified for mobile microscope sufficient to promote integration and acceptance by rural clinicians and physicians and veterinarians for diagnostics. And so that's kind of the space we've been at in this early part of the project is as we're talking about redesigning, redeveloping, and expanding devices like the U-phone, are they thoroughly designed? And what does it mean for them to be thoroughly designed? Is not clear yet. And so a critical challenge is defining engineering requirements around existing clinical workflows to try and complement, integrate, and modify where needed existing workflows. So I guess I'll keep chugging along. So this is kind of I'm going to go into some of the more clinical implications of the project, but does anyone have any questions or comments or thoughts as we keep moving forward? Yes, Dr. Reed, I've got a hand raised.
Um, yeah, so I don't think I thought about it until you just described it this way. And, I like building machines. And so I'm tempted to say, well, let's just build a microscope. But first of all, the open flexure microscope may be what, you know, may accomplish a lot of the things that we need. But you're now raising the point of something that plagued the emergency ventilator problem, which I was involved with a little bit on a global scale, which was that even if you have the equipment, even if you could just wave a magic wand and give everybody an open flexure microscope who wanted one, you might not have trained staff who would be able to use it. Now, of course, at Public Invention, we're not here to conjecture about things. We're here to solve problems, right? But I have noticed many times acting as a judge for the global health design competition at Rice University that some of the best projects involve training in one way or another. Um, Now, I therefore wonder and put forward the possibility that maybe what's really needed, even though in some ways it's less nerdy than building a microscope, is simply to build a microscope training system. You know, and when I was a boy, I was very lucky. I had a Sears microscope that was very high quality. It only had one eyepiece, and from an image point of view, it probably wasn't great. But one of the great things about it was it came with a set of 20 already made slides that included blood cells. Um, like I remember it had a preserved dandelion seed, and a variety of other very interesting little things for a young child to look at and understand under a microscope. Although it would seem to be less ambitious than some of the other things that Public Invention has done. Maybe we should just think of building a training system.
Yes, and certainly, as I think about it, and as I started working on the project, and I was thinking about, how might I encourage adoption or use or assist in that integration process. It's like I don't like I know how to use a microscope. Certainly don't know what a pathologist does, or I don't know how a pathologist communicates with a clinician. I don't even know when a clinician triggers a diagnostic process that includes a microscope. I mean, there are certainly going to be clinicians who say like, I've never used a microscope. I've never worked with a pathologist. Like that's not what I do. Um, so yeah, like how do you develop infrastructure, training, onboarding, acceptance? And thankfully we have, this isn't one of those COVID type of problems where it has to be solved right away. And there are, as you mentioned, there are great existing solutions like open flexure microscope. But almost on, like you said, on the level of like, okay, has anyone studied how doctors really think about this and what kind of impact it can have on the field outside of, I mean, sure, let's say it can, detect cancer 90% of the time. And the question is like, who is doing that? Like, is it the people who made it? Can a novice do it? Can a novice utilize it? And that, I was inspired by your early guidance when we started the project of we can maybe make the best microscope, but like if it can't be used by anyone, then it's, it's not usable, so it's not useful. So um certainly trying to get that question of we want to, and it may be the case that OpenFlexure is the best usable solution for clinical microscope diagnostics. In which case, I think our project should be to capture that dynamic and characterize it numerically. So, yeah, I think we're headed in the right direction of trying to answer some of these unanswered questions.
Yeah, well, before you answer Megan's question, let me just say public invention is always in favor of cooperation over competition, right? We don't want to build a new microscope if the open flexure microscope can do it. And I'm sure there are ways to improve the open flexure microscope or improve the training system around it that would be well appreciated. Thank you.
Yeah, yes. And thank you, Megan, for your point about, and it's nice to have, oh, I think that was Victoria's point about, and I think it's really nice to have people in our project who have clinical interactions or work with clinicians. Because really I've kind of been in my mechanical engineering world for so long that, and I think a lot of people have in our space. So it's great to get that kind of perspective. So thank you. And I was able to talk to Julian this week, and I'll talk briefly about it later in the presentation. But they're working on a future application for bovine tuberculosis assessment. So I think we're kind of tracking and almost going to meet in the middle. So I had the thought that I might just send him, mail him samples, or have him send me a device, and we'll definitely see what can be done. So I think there's certainly going to be some interaction with him. He invented the microscope and the interaction with their project. So I'll keep chugging along. So we talked about, we should define engineering requirements around clinical workflows. So what are clinical workflows? So in broad strokes, I'm not a clinician, but I've done, some Googling and some reading. In broad strokes, you start with a clinical indication and the selection of a site. So would be part of oh, so I'm sorry, Megan. I don't know if I missed a question from you. Do you have a question? I don't mind going back.
So it wasn't too big, so I was just gonna let you keep going. But essentially, it's kind of what you were ending on that final point, which was what benefit do you think bridge programs could offer for projects that ride the border between two fields like this? And are those experts truly beneficial when designing these products?
Yes, thank you. Great questions. And those are the questions that, as an engineer, I'm afraid to ask because it's like, oh, I can't do everything. You can't keep everyone happy. But I think, we're at a stage where it's not such a critical problem, and it's also somewhat well addressed that it's now at that stage for maybe refinement is the project needed and, characterization of existing solutions, um, rather than starting something from scratch. So hopefully, we're at that stage of bridging initiatives and assisting existing initiatives. Yeah, so what are field clinical workflows? They might start with clinical indication and selection of a site for sample extraction and so forth. Uh, so then we have our sample extraction and kind of labeling, transportation, processing. I guess that's, yeah, and preparing a sample. So we've kind of gone from a patient presents with some symptoms, or there's some desire to characterize a biological sample, to extracting and preparing. And then kind of the microscope side of it. So the technical element of obtaining, how do you obtain the best images of such a sample? And then acquiring either by some digital or intelligent means or by direct observation, key features of the image in the slide. So analysis and interpretation, we talked about how that could even be a distributed process with multiple clinicians being able to contribute. So this kind of pathology um, step, and then documentation reporting. Let's see if I have, and yeah, and then there's a review and collaboration. So patient presents with something, or the farmer has provided an animal and talked about some symptoms, down to when does this need to get to the next analyst and the next clinical step. And so kind of everything in here are the steps that we're trying to say, how does a field microscope make these things possible, make these things easy to do, reduce the barrier for entry for non-experts to do these things. So that's our goal. And so we're writing a systematic literature review on this topic of how do existing open microscopes align with clinical workflows. So we're segmenting diagnostic workflows, kind of like the previous slide was a segmentation of broad workflow, but we're segmenting specific design workflows for several dozen medical diagnostic pathways. And so we're mapping standardized steps. So, obviously there's the step of a patient presents with X symptom, but there are also some really fine steps. And so the question is, how fine do we go in identifying steps that might cause a hang up in the usage of a microscope? So that's part of our process. And then we're assessing the compatibility of existing microscopes like OpenFlexure. And so my hope is we look at closed, open source, mobile, and benchtop microscopes. So, and there are even, broader categories than this in terms of existing microscopes that are hoping to compete in this space. And we're kind of about maybe 3/4 of the way through, but I guess that last quarter is the hard, the 90% of the effort. So, and so we're planning to submit this to the Journal of Microscopy with the deadline coming up in September, I think. And so I was able to talk to Julian about OpenFlexure and able to see it in action. And I think it's a really great platform if we can collaborate with them. So, Dr. Reed and Megan know that we have a smaller kind of off-the-shelf microscope. If we're able to use that as kind of a step one and then send samples to Julian or to his team as a step two, then maybe that's some pathway for collaboration. Or again, he sends us microscopes and we're able to kind of utilize a broad scope of tools. That may be another, or maybe he sends me some tuberculosis samples or something. I don't know. But yeah, I mean, and it's great because he's, from the UK, so there's kind of that element to navigate as well. But I think this is part of our scientific process where we are now. And so another step that we're looking at, in addition to doing some of the literature review and industrial systems design elements, is kind of some of the more practical aspects of the project. So I mentioned earlier in the talk that I've recently moved to a rural community in Alabama. And part of this was personally motivated, goals about my life and my family and so forth, but also I'm really motivated by a question I've been having, which is how ready is rural America to embrace open source holistically as kind of the first step in any oh, there we go. Let's see. Yeah, great. Yeah, thank you. Great point, Lawrence. Which is that, yeah, and I'm presupposing a lot in even my selection of who might be interested in such a thing. And so with my access to a rural community in Alabama, I've noticed that people are interested in open source. They're very interested in the right to repair, the right to understand what's in their technology. They're interested in, not participating in economies of scale which don't seek to serve them and don't have their best interests at heart. And I think they're interested in participating with economical cycles where people have their interests at heart and are trying to, trying to support them and care about them and want them to succeed and aren't trying to make, a million microscopes, but even if we make 1,000 microscopes, that would be a success to us. Yeah. And so I've specifically thought about how I might go about seeing and testing the waters in a rural community. And I realized that actually clinical workflows have a lot to do with agronomy workflows. So it's kind of like not veterinary science, though that's involved, but literally farmers constantly diagnose their animals and constantly take steps to treat them to improve their own yield in their farms. And so they kind of act like vets, their animals are products really, but they're also medical patients to them in certain steps. So, we talk about clinical indication. So these are the steps that I had previously mentioned in clinical workflow. And so that could map to agronomy in terms of, identifying and seeing what a patient presents with, whether that be a plant or an animal, right? Extracting samples, so collecting biological samples from animals, which may be many of the same samples like blood, stool, saliva, skin samples that we might collect from humans. And then, of course, we talk about very highly scalable tests, which may be done on plants and so forth. Sample preparation. So again, if we're using animal samples. They should, I should do some research before I claim this, but I expect they can be processed in the same way that, human biological samples can be processed. So I'm going to skip through some of these just for time's sake, but the broad point here is that the same steps that a clinician might take who is doing some kind of clinical diagnostics, we could have farmers do that, in their practice. And that may be a way for us to do some industrial systems experiments with folks who might be open to open source and may want to start participating in that economy. So that's something that we've been looking at. And so this is kind of a big spread here, and I'll go through each of these columns one by one. But we've been talking specifically about an example of chickens. And so there's the option of looking at biological samples during the growth, processing, and post-processing of chickens as meat, but also eggs. Right. And so that's kind of and in both cases, actually, interestingly, you need very little regulatory approval anywhere if you own the animals and if you're not intending to treat the animals based on the data that you collect. So, I need to confirm for sure. I need to talk to a lawyer or a politician or something. But from my basic research, you can collect almost any of these samples, do analysis, and publish it for chickens without any regulatory approval. And so that horrible, statement aside, as my disclaimer, there is some scientific questions to be asked. So I'm going to pull into there's a rearing phase of growing chickens, which might lay eggs. And so in that phase, we might assess samples like amniotic fluid and yolk contents for eggs we purchase to breed and hatch chickens, kind of the stage one of the poultry process. Then during the rearing process, we can check blood samples. And so I guess before I continue, something I have been thinking about is how in any clinical setting, but also farmers, how they might process samples so that they're actually representative of the larger sample that they're trying to characterize. So, for example, feathers, like an individual feather may not be representative, or an individual tick infestation site on a single animal may not be representative of the health of the animal as a whole. Whereas if you wash the chicken, the wash water that comes off of the chicken may be more holistic representation of the health of the chicken. So there's some cool questions to be asked there. And then I'm going to skip through some things because I wanted to get to this point, which is there are some really interesting questions, not only about the egg laying capacity of a chicken with regards to a test that might be done that's similar to a pap smear, but also interesting things that might come out in terms of somewhat less economical, but health related tests as well. So a chicken pap smear may be a really interesting analog for clinical workflow in humans. So yeah. See if there's anything interesting here. And so some of the interesting things I've noticed is there may be interesting samples of biological material that may be very easy to extract and highly representative of the health of the animal, like rinse residue or just grinding up eggshells, right? And processing those as um, uh, biological material characteristic of the health of the animal. So yeah, I think there's a lot of cool questions and experiments to be done with chickens, which may be very easy. Yes, I see a question. Go ahead.
So there was a study that came out this year maybe, talking about how dust plumes from H5N1 infected farms were carrying the virus to uninfected farms. So I think there's enormous value in doing ground and just general water in the area sampling, not just in the poultry area specifically. I would say air sampling as well, but that's a whole nother field for that. Because like a wider range of data collection, I think, is important, because the culling of the birds is a huge problem in rural America, because that is people's livelihood. It costs them money, insurance, all that sort of thing. And I see this technology as a way for farmers to have some data ownership and have autonomy, like being the first line of defense. You know, I'm the first person who's looking at this and starting a data stream. Because what happens is the inspectors come in, the samples get sent out, and then the farmer gets to report, like, okay, you got to kill 500 of your birds. And they're like, okay, well, I guess I trust you. So I think the farmer being the first line of data collection could start more of a trustworthy science process, because there's too much of, you know, I have to do this because the government told me, and I don't know if it's correct. And there's all sorts of reasons why someone might mistrust, you know? So this democratizes and reduces a lot of data gatekeeping, I think.
Yeah, thank you. You know, I've kind of been on the cusp of just straight up interviewing people out there to see what they think. And we've talked about this, you know, just to get a sense of what they think about open source in general, and open science. And what the implications might be for their day-to-day lives. So, yeah, thank you. And I hope we get more people like you who have these, you know, who are well read, you know, in the space and might be able to talk to us about this. So thank you very much. And so how do we plan to do this chicken project, right? So it's not really, and I certainly don't want to become not a microscope project, but a chicken project, which I think could become a whole problem. Um, and so a big part of it is I think we'll hope to work with people who already have chickens and try and get into those workflows that already exist. So let's see what I have. So we did purchase the Sandmark iPhone-based microscope platform. It's about $200. The magnification is not great, so it's really more of a macroscopic imaging platform. So if we were to use it for things that we might do in the field, so the plant imaging, so you know, trying to find kind of visible defects and visible parasites in plants and so forth. And so we're kind of trying to see what we can do with this for now, because commercially available, it was obviously closed source. It's commercially available. We could. It doesn't take a lot of effort to build it. It comes pre-assembled and so forth. And secondly, we would like to work with folks like the Open Flexure microscope, and in general, the benchtop, you know, robust, self-focusing, Wi-Fi enabled platforms, which may be able to do true microscopy. And then our phase 3 would be to integrate in veterinary environments, then maybe in urgent care clinics, and then maybe in rural public institutions. So kind of to Victoria's point is being able to allow communities to take control of samples, maybe have scientific co-ops, which you know, are able to serve a community reliably and in a trustworthy way. And yeah, so that's the large scale plan we have, is to use the Sandmark microscope to do some science, at the same time do our kind of industrial systems study of workflows, and then maybe kind of in the fall we'll start to really dig deeper into what you know, kind of high grade traditional microscopes can do in the poultry science space. So I think that's about all I've got. So does anyone have any questions or discussion from here? Yeah, go for it, Dr. Reed.
Well, I certainly would like to have other people ask questions, but nonetheless, I wonder, the open flexure microscope may be the best open source solution that we have today. Does it have a supporting ecosystem in the sense of software modularity to provide diagnostic tests associated with it as we've conjectured? Maybe we're behind the curve on that front as well.
So it is possible that they have that in the demo that I saw with Julian. It's not clear that the microscope, and it's possibly the case that they don't want to take that step for liability reasons and so forth. It's clear that their platform in theory could do that, but it is not clear that it does that right now.
Okay,
The
thank you.
question, yeah,
Uh,
there's a question about PCR testing. So an interesting point about PCR is it's the gold standard for bird flu detection. So I would like to get that question answered. So I'll look into it. Thank you. Um, I've got, oh, this is interesting. I will look into this. Thank you. Yeah, thank you, Lawrence. I'll look into that microscope.
Avinash, when this is up on YouTube, you should put it on your LinkedIn, and I'll reshare it and tag a whole bunch of people from the CBMS workshop.
Yes,
Because
and
Severin Cazal, especially in Santiago de Chile, would really like to see this project.
yes, thank you very much for connecting us to that community. And I will definitely do that. Thank you. I'll try and slowly scroll through our slides to see if anyone's got any questions that come up.
I think Megan has a question.
Okay, go for it.
So something in the back of my mind is the consideration of having too many masters, but not enough, I guess, like hands to reap the crops. And I say this abstractly because I'm trying to get at the point that with poultry science, what would be something we could truly tackle? Because as a teammate, this is a consideration we're doing by finishing out the literature review. But I ask this because I'm kind of curious after looking at the presentations in New York and a little bit of the knowledge you've gotten in the last week or so. Um, what do you think truly we could target with the capacity of the existing open source microscopes for poultry? You might, as you said, like you need to look into it further, read papers. But where are you leaning? Where are you sitting right now for like what your target would be?
Yeah, it's true. I am not a scientist, so I'm probably going to make claims that are wrong. So what I mean is I'm not a, veterinary scientist. But this question of how well does a pap smear for a chicken map to a pap smear for a human in terms of the health impact that resulting diagnostic decisions may have. So one simple thing is apparently they control large populations of chickens with light. They're light guided. So if you turn on the lights, they will just eat nonstop, and they won't eat if it's dark. So in order to improve the weight of the chickens in darker, less sunlight, months with less sunlight, they'll just artificially, they'll add a few hours in the morning and the night of artificial light. Um, but that can cause things like stress, and also it might be worthwhile to consider the result. Oh, we gotta go check. Thank you. Yeah. So the question of the pap smear might actually be really indicative of stress related to light exposure. It also may be indicative of how many eggs a certain chicken will lay at a certain period of time, which might be useful for guiding things like how should you do your light stimulation, how should you distribute those resources to the chickens who may benefit from them, and not to those which may be harmed by them. So for me, I'm thinking in terms of the pap smear experiment, using a more robust microscope, like maybe like like the open flexure microscope. I mean, certainly the numbers always slip my head, but Sandmark, its magnification is not sufficient enough to look at blood cells. Or at least I don't think so. I got to confirm that. So we also probably wouldn't be using it for our pap smear experiment. We might start with plants, right? And be looking for aphids and so forth. But my hope is to really do something more significant where we're aligning with a classical biological sample that is relevant for human diagnostics. Is that kind of what is that interesting to you as a
Definitely. I think
task?
that there's benefit there because I don't think that it's very clear. I don't think it's well taught on or understood between the two. Like if you go to med school, if you go to any type of pre-health program, I don't think they really dance that line between like when animal testing is sufficient enough to have a cross parallel to human testing clinical trials. They just use it to rule out risk, and then once they get to a certain level of risk, like you've mitigated enough risk, they'll start initiating clinical trials, but they can't truly say that one will lead to the effects of the other solidly. So I think finding that comparative, like by proving it with this sample, you have a higher chance of having successful drug delivery or, I don't know, treatment for this vector uptake through a plasmid. It just really depends. I think if we elucidate a little bit more clarity, that is definitely beneficial within either realm.
Yeah, I want to keep talking about that, but I see other Dr. Reed, do you have a question or a comment?
Yeah, this relates to my previous comment. So here's a problem that makers and researchers like ourselves have to face, right? Um, let's now say that suppose we could give an open flexure microscope to every uh, clinician in Alabama, and that we had good training materials for a wide variety of disease conditions which they would be able to assess. You know, if they're still completely time constrained, that is, their workload doesn't allow them the time to do this, then they might still, in essence say, "Oh, well, that's nice, but it doesn't change the way I do my work," right? It seems like what we need to seek is a way in which this relatively inexpensive, relatively adaptable technology could do something that they're already doing, but save them time in doing it. You know, of course, that might not be right. The alternative is we can say, well, look, we're opening the door to assessing disease conditions which you can't assess today. But if but then the person has to make a pretty complicated decision, right? Like I've got 8 hours in a day. I'm going to take 1 hour out and look for diseases I wasn't looking for before. That will no doubt detect some treatable diseases, but I'm giving up an hour of the other work, which was previously deemed to be very important anyway, right? Um, I don't know if you're going to be able to find anything like that, but I think we should keep our eyes open for it. Thank you.
Thank you. Victoria, do you have something to say there?
So we have to remember that people looking in microscopes is very niche departments. It is lab doing a manual differential. It is patho. It is maybe pharmacy once in a while, but mostly lab. You send the fluids to the lab, and that's where microscopes are used. Microscopes are not used by emergency, surgery, cardiology. They're just not. The interpreters of the fluids turn the pictures into data, and then those numbers are interpreted and decisions made by the frontline physicians or the specialists. So and we also have to remember that this same design is being very eagerly commercialized and privatized. There is a market for this device, I think, far beyond medicine. Otherwise, there would not be a commercial interest in it. So I think that probably the animal side of this is the most probably the most apparent, where you're going to get a lot of data, which will probably open doors to human side. I can see this probably doing very well for a waterborne pathogen. You got to get something like this out there and people talking about it and using it, and then suddenly you're going to find a lot of applications that you can't even imagine.
Definitely. Yeah. Yeah, you're absolutely right. I think there are a lot of, I think it's likely that the conversations we have with clinicians, the conversations we have with, and our work on the lit review will reveal some things which may redirect our approach. And I think one of that will be an answer to the question like, does a, first responder in any, in anywhere in the world need a microscope? And the answer is almost certainly going to be no. So if we answer that question, that will be, I think, worth knowing for sure.
So I'd like to talk to Victoria about that for just a second. So in rural places in America, does that mean we shouldn't be asking, we shouldn't be considering this as a point of care device, but as a way to improve the efficiency of a pathology lab?
And I'll make a small addition in that there might also be the goal of self-testing. So kind of robust like self-blood testing and so forth. That might be a worthwhile topic to talk about also.
So if you go to, I'm trying to think of an urgent care or a doctor's office in very rural America. Those structures are going to be associated with some sort of health system through which they have access to increasing levels of services. There's going to be some point of care and on-site lab services. Like you can run some eye stats and basic blood tests, but they're not done with a microscope. They're all blood and analysis and centrifuge and things like that. And then more complicated tests, they're sent out. They are, mailed overnight. They go by courier, and you get results in 48 hours. So if I don't honestly really see this being used like in family practice. I really see it more like waterborne pathogen, animals, zoos, maybe. Definitely research, because the more field research you're doing, the smaller and lighter you need things to be, because if you're on the side of a cliff measuring bacteria in seagull guano, you probably want something small in your backpack. So I see this being a really big boon for a researcher. And there may be, medical applications again or medical locations that need this that we don't even know about yet. And I do see it as being like being an open flexure ecosystem, because you could, I'm sure, make the phone talk to the microscope in some way, and you could upload data to the flexure, because I know it has a USB port for transmission and stuff. So I see us here maybe having a smaller, lighter open flexure.
So let me ask you this, Victoria. Let's try to work within the existing structure in which there's a point of care clinic, and then there's a pathology lab, which is far away in time and space, right? Now, could we get the pathology lab to operate only as telemedicine based on images taken at the point of care in order to speed up diagnosis without compromising um, the the skill of the pathologist, and without changing the structure that that's the path lab that knows how to deal with the images. Um, I suppose that would mean these issues of, thank you, Larry. I suppose these issues of microscope slide preparation would have to be addressed in the field. But if we did that in the field, would that might be something that might be very welcome um, to even to the pathologist, because they wouldn't have to do the slide preparation.
Well, what you're talking about is, can we have faster, smaller litopatho? Do we have a tiny little patho in our pocket? And it's not really that simple. And to any pathologists who are going to listen to this YouTube video, I really respect your profession here. So you have to remember that for patho to interpret most samples, they have to be prepped a certain way. You have to have stains, you have to have certain preparations, and you have to have the skill to interpret. So I'm not sure how sophisticated this system could be. It could be that, yes, this could be a data collection point, and images could be sent to patho, but then who's going to read that? Who's going to read that? And who's going to take whatever you send as, this is the highest quality sample I can get? Probably they're going to say, okay, I think it could be this, but I need you to send me the actual thing because I need to prep it myself. You could get maybe a preliminary reading, but if you're 3 a.m. needing a patho read, that's not a very emergent thing. Patho read takes a while. I mean, I've had a patho read done for myself. It took a week and a half to get back. So unless you've contracted with a pathology group in Australia, like radiologists do, to read American x-rays during their day and our night, I don't think you're going to get a faster read. And the pathology world these are very passionate people who love microscopes and reading samples, and they are usually by themselves in a room in the basement of a hospital, just over a microscope all day long. That's what they do. That's what they're good at. I don't know how to make them faster, and I don't know if this device is what can bring us more patho readings.
Megan, do you have something to add?
Yeah, no, I think she hit it right on the head. Like, for just a quick example, I had to get certain vaccinations updated for clinical stuff. And in that, I had to get like a basic TB test. You can do a skin or a blood. The blood is supposedly faster. It took about a week and I think an extra day for them to follow up. And that was at a local clinic in the city. You can only imagine if like rural hospitals are further in between, most cities. Like my friend, she goes all the way to Austin for her OB-GYN. She lives in Giddings. Like I think it really speaks volumes of like how many options exist for pathology labs, how overloaded they are, as well as they take it very seriously, and they need like legitimate samples in order to confirm things, right? Like it's really hard for them to confirm stuff if the sample is prepped by an outside person who might not have the licensure to do that, or the laboratory environment to make a solid sample. Um, One thing I think about is like the detergents, because sometimes there are chemical agents they use in the laboratory in order to break down certain samples, better imaging. It could also be the temperature. Like there's various laboratory environments, and that's like requirements that make samples more readable and more consistent, more reproducible, stuff like that. So I can't speak for a pathologist because I'm not in that field, but I have a lot of friends who are in pathology as residents. And like she said, they do take it very seriously. They do enjoy the stuff they're doing as well as their like lab work. Like I have some family members who go into that, like as technicians, laboratory technicians that process it. And it is a little bit stringent. Usually you're not trying to get a sample overnight for ER cases, like she's saying, like you're not going to test for a stroke that way or something where it's an immediate like risk factor. So I think that in a way that when she made that, that came back to my mind and made me think, I think the benefit for sure would be a long-term gain, something where you're trying to monitor like almost like batch testing for manufacturing lines, like compliance testing. It would be that before poultry or for animals, I would assume, where you're taking live samples to validate like the generation that you're growing or the feedstock for the supply for a quarter or some time frame where you have to retest it and retest it, retest it. That makes more sense in like a rugged kind of case. Um, but my mind also goes to if you're looking at like organisms within an ecosystem, a good way to differentiate speciation under pressure could also possibly be that if you can look at reproductive, like developmental stages of like frog eggs. Um, I don't know how beneficial that is. I'm not a marine biologist or any type of biologist, but I would definitely think stuff like that in rugged environment would benefit more than healthcare applications. So she hit it right on the nail.
I think our next
Yeah.
step might be to actually sit down and interview some pathologists who, you know, are maybe a little bit outside the box, and most of them are. Pathos are really amazing, amazing specialists, and see what they think about this. And, you know, is there something we don't know about that could be an application for it?
Thank you. I will add that to my to-dos. I think that is certainly part of that should certainly be our next step, is to talk to people who might be our users. And if I can, I'll try and invite Julian to those meetings as well, because I imagine he'll be interested in that topic. How are we doing on time? I think we did decent on time, I think.
The only like rural medicine application I've heard randomly is rural Australia, where very innovative, small, 3D printed, unusual type things are maybe able to be trialed because rural Australia is that. It's very, very rural. I don't really have connections there, but that is one place in the world where something like this, once it has enough legs underneath it, might have some medical application that we can't anticipate yet.
I'm going to stop sharing just so I can compile some notes on the side, but not to cut off the discussion.
Okay. Well, this might be a good time to stop this here. Thank you, everybody, for attending. And I hope if you're listening to this, you'll join us in the future for our third Thursday Inventors Gathering. Thank you, everybody, for attending. I'm going to stop the recording now.